and it's different kinds of data. For common diseases, it's very difficult because the genetic correlates of common disease can be only minimally penetrant. So they may involve very many genes, but different sets of genes for different people and so on. Yeah, statistical genetics is an interesting science. It's not easy.
So when you make a full map of signatures of a gene that has some causality disease, do people continue to explain why that gene can do that disease? Or just observe the correlations?
Yeah, so absolutely. So they'll do things like they'll look at they'll do studies first in cell lines that are derived from tumors to see how essential those genes are in critical viability studies in the tumor. So they'll knock that gene down, they'll stop it from being expressed, and see if those cells die, to see whether it affects that. Then they go on to mice, so they'll do in vivo studies in mice. Mice are used a lot in these kinds of studies. The next step in these kinds of studies, you can do things in mice that don't necessarily translate to humans, but that's the next step. So yeah, that I can't do on my own. I don't go into the lab. I have good collaborators that go into the lab. I mean, the other thing is that some of these things are going into the clinic. I didn't speak about it, but we did a study on lymphoma a number of years ago where we found a particular gene was overexpressed in poor outcome patients in lymphoma. And again, there was an inhibitor for that particular gene, which was used to treat diabetic retinopathy. That actually went right into a clinical trial. And so that was the problem. Yes, no, your questions are not so.
There are no statisticians check if the work is a Bayesian model.
Yeah, it's a statistical model which also takes into account priors. It's a statistical model which also takes into account the priors. The prior probability.
So
That's the short answer.
So they are the technical approach priors.
Yeah. Yes.
In your slide, I saw something strict when you compare four models, but in the first two models, the performance on training data is sometimes better.
Because remember the features for those two models were not determined by the training data. They weren't learned on the training data. They came from, that's a good point. I thought I had mentioned that, but maybe not. So the histology and the metastasis are clinical attributes. They weren't trained. Okay, so we just use them out of the box because if the patient has metastasis, that's associated with poor outcome. So we didn't train them to do that, right?
Nice.
And c-MYC activation, that's the poor outcome feature there, also was derived from the literature. So that's why you see that funny thing in models A and B. But then in model C and D, we didn't train. They were trained. Good question.
In the past, I also done some experiments on biological data using machine learning technique. Actually, it was protein interaction prediction, yes. But my problem is even if we can collect a huge volume of data, extract many features, related features, and build some model to predict that pair of protein they interact with each other. But the problem is if we work on computer science or machine learning only, so we have no way to know that the pair of protein they really interact with each other.
I agree.
So
no, you're absolutely right, and that's exactly why we're not just using computational methods to get these gene signatures that I was talking about, but why we're trying to derive them experimentally and all that. I mean, I really meant that last slide, which is that, let's go back to my religion. This is my religion, you see. Biology for computers, well, it could as easily be biology. I think it's up there, biology for mathematicians. I mean, I don't think you can do good, this is my own opinion. I don't think you can do good computational biology or mathematical biology without really understanding the biology and without really getting involved with the biology. I mean, you gave a perfect example of that.
Any question? Another question, they thank you.